Good afternoon, everyone. Nice to see you all. You haven't seen my face before. I'm Dr. Manuel Copas. I'm a senior lecturer in genomics, and I am the sort of co-leader for this module, molecular genetics, and we're going to have a few lectures together this week. I'm going to present some of the basic concepts for genetics, and let's see how it goes. I'm very happy. If you have any questions anytime, please stop me. I love having discussions about the science and the contents. Are you all able to see the screens properly, or is this light annoying? Can you hear me at the back? Okay. So, have you all heard about Gregor Mendel? Yeah, we're going to be talking about Gregor Mendel today, and really appreciate how in the second half of the 19th century, someone in an alley was able to develop what would be the concepts that a couple of almost 2 centuries later we're still using. So I find that completely fascinating. Anyway, so we're going to learn about the theory and the concepts that Gregor Mendel suggested 170 years ago. So I'm sure you all know what a chromosome is. Can someone remind me how many pairs of chromosomes do we have in the human genome? How many pairs of chromosomes do we have? And why do we have pairs of chromosomes? Why not just having one copy instead of 2 copies?
Yes, we have 24 chromosomes.
Sorry?
24 pairs of
23 pairs of chromosomes. We have 22 pairs, which we call autosomes, and then we have another extra pair. Can you tell me what the extra pair is? Something to do with sex? Sex chromosomes? We all know that the number, we call the chromosomes 1, 2, 3, 4, 5 up to number 22. Can someone tell me why they're called that way? Is this siesta time? I'm Spanish, so I should have a siesta as well. Come on, can anyone tell me or think why we would call the chromosomes with those numbers? To identify the location of genes, okay? So they are called that way because of sizes. So chromosome 1 is the biggest, chromosome 22 is the smallest of the autosomes. And there's a story behind. Can anybody tell me or suggest why the sexual pair are called X and Y and not those numbers? That's a good guess. Any other guesses? It's because they were weird. They had a sort of strange behavior, and so the people who discovered those chromosomes, it was kind of an incognita in the same way as we have equations x and y. So that's why. So it comes from that association to be a sort of incognita in mathematical equations, because their behavior was slightly odd compared to the others. Okay, fantastic. So chromosomes really is what we see when we look under the microscope and try to see the actual genetic material of eukaryotic cells. Can someone remind me what an eukaryotic cell is? It has a nucleus. Fantastic. And what's special about the nucleus? It contains genetic material that most of the genetic material. Can someone tell me where there is material as well, genetic material, apart from the nucleus? Where exactly in the cytoplasm? Ribosomes, no, it's on the mitochondria. So sometimes people call it the mitochondrial chromosome. So we have the vast majority of the DNA is in the nucleus, and then we have a very precise number of nucleotides bases in a mitochondria. It's around 16,000. A bit more than 16,000 nucleotides, and you always have that number of nucleotides. But can someone tell me how long is the longest chromosome, chromosome 1? Do you have an idea how many nucleotides it might have? Any guesses? Is it millions? Is it hundreds of millions? Is it thousands of millions? How long is the human genome? How many bases does it have?
4 bases.
Sorry?
4 bases. The human genome, 4 bases.
It has 4 bases, but when we take all of the length of all the nucleotides that make the human genome, how many bases do you think there are? 3.3 billion. Right, so it's not a small number, and if you put every nucleotide base in all of the chromosomes in a sequence, a font of 12 point, which is the default for Microsoft Word, you put that all in a single string of letters, it would be 7,000 miles long. Okay, and we have that in every cell of the trillion cells that compose each of our bodies. So now we begin to have an idea of the scale of this information, and that information, all of it is, or most of it is encoded in the chromosomes, which is the hereditary material, and that's what we inherit. So why do we have 2 copies of chromosomes? Can anyone tell me? Sir?
To share the parental.
To share
The parental genome.
to share the parents' genomes. Yeah, that's about it. So we have one chromosomal copy comes from the mother, and the other one comes from the father, biological father. And basically, that's why we have 2 copies. So we can't function with just 1 copy. We need to have the 2 chromosomal copies, and we're going to look into that mechanism in this lecture. I'm sure you all know what a gene is. Yeah, so a Can someone tell me, apart from that definition I have here, a region of DNA that controls a specific hereditary trait function, what is a gene really in the chromosome? Can someone tell me what it is physically? Sorry? Sorry, I'm a bit deaf. 3 colons? Okay. So this is not my favorite definition of a gene. My favorite definition of a gene is a segment of DNA that encodes for a protein, usually. Okay? There are exceptions to that, but I'm just going to go with that definition for now. Okay, so happy so far, right? Any questions? Cool. So let's look at this person. So Gregor Mendel, he was actually a monk at an abbey in Brno, which is today is the Czech Republic. Okay, so he was a priest. You can see the monastery where he worked, and basically he was interested in counting, and he worked on peas, Piscis sativum. He studied the variation in pea plants using the monastery garden through cross-fertilization. And peas are easy to cultivate and easy to handle, and they reproduce a lot. So the genius of Gregor Mendel wasn't just on the meticulous collection of data that he performed. That was in itself wondrous. But also, the genius was that he used the right species to be able to conduct controlled experiments where he would be able to then count and infer specific patterns around how the different traits are inherited. So he developed 3 laws of inheritance, which we're going to look into, and this has to do with how genes are transmitted from parents to offspring. And even though this was done with peas, actually it applies to every sexually reproduced organism. So it has a huge application, not just to peas, but also the same laws apply to human. And here we're going to look into 2 laws, the law of segregation and the law of independent assortment. So how is inheritance? How does it work at a molecular level? So you may have heard of something called meiosis. Have you heard that term before? Right. And so that's how it happens. And I mean, a little bit about peas. So what's so special about the sort of study of peas? So they're easy to cultivate, as I said earlier, and they can grow in large numbers in a small space, and the fertilization can be controlled. And then there are many different varieties with different traits, different phenotypes, as we call them. So have you heard the word phenotype? Yes? Cool. So how does this process work? So the way he did it, so he removed the estamens from the purple flower, then he transferred the pollen from the estamens of white flower to the carpel of purple flower, and then the carpel was matured into a pod, and then planted the seeds from the pod, and then simply just examined the offspring, looking at the different traits that he was looking into. As I said earlier, it has implications because the same laws apply to every sexually reproduced organism. Here, for instance, the same law, if we have an albino man and a normal woman, the first generation is not going to show, and that's probably because it's recessive. And here you have a normal man with an albino woman. They don't show the trait, but they carry the trait, right? And then if you have from these two first generations a crossover, then there's going to be a 25% chance that you're going to have albino offspring. So the same mechanisms and the same proportions as we see in bees, they apply also to human, for instance in this case to albinos. So we've mentioned already what a genome is. So a genome is really all of the genetic information of an organism. And actually I disagree with that. It's not just in the nucleus, because you can also have genetic data in the mitochondria. It says here may also include the mitochondrial genome. So something really important, alleles. So I'm going to be talking about alleles a lot of the time. So looking back here, we have, let's say, two alleles, right? We have two alleles, and an allele is a variant of the same gene. So here the assumption is that we have one gene, which is the one that is going to condition the color of the skin, the color of your hair, right? It's usually the melanocortin receptor 1 gene. And depending on the different type of genetic code that may have the same gene, you're going to have different traits. And it is this difference for the same gene that we call an allele, okay? So you can, in this case, how many alleles do we have? We have two different alleles, because we are assuming that there are two different versions of the same gene, and depending on which version we have, we're going to have a different phenotype. Does that make sense? Any questions so far? Am I going too slow or too quick, or just okay? Okay? Cool. So an allele, if we look at an allele physically, it may be just one nucleotide change or more than one nucleotide change. And I mean, you could call it mutation, but the mutation is slightly different, because an allele always makes reference to the same type of mutation, whereas a mutation can be any kind of change, okay? So that's important to know. Right, have you heard of homozygous and heterozygous? So this we call it zygosity. So zygosity only makes sense in the context of having a diploid genome. Can someone tell me what being diploid means? What's a diploid organism? Correct. What's a hexaploid organism? 6 copies. Can someone tell me an example of a hexaploid organism? Wheat. Wheat is hexaploid, okay? So you find that in plants you've got weird combinations of, not weird, but complex number of ploidy, okay? So in all of the organisms that we're going to look at, they're always going to be diploid, okay? So there's 2 copies for each chromosome, except for the sexual chromosome, which has this odd XY different sizes. But there are also many interesting mechanisms around the sexual chromosome. We're not going to go into those on this lecture, or in the future even. But here we're just going to assume that we're going to be talking about the autosomes, okay, which are the ones 1 to 22 chromosomes, which are identical. Well, no, they're not identical, they're homologous, okay? If they were identical, then they wouldn't be homologous, okay? Homologous is that you have one copy coming from the mother, one copy coming from the father. They are similar, but they are not identical, okay? Because the mother and the father have slightly different variations, slightly different alleles actually. So here we have 2 alleles, okay? So we have an allele, a red allele and a blue allele. So if we have the same allele in both chromosomal copies, then we have homozygous. We have a homozygous allele. If we have different alleles, a different allele could be something like, in this position in chromosome 1 you have a A, and in same position in chromosome 1 in the other homologous copy of the chromosome you have a T, right? So you have, let's say, AT. So that instead of having a AA, right? I'm just making this up, but hopefully that makes sense. So that's what a heterozygosity is, when we have 2 different copies of the same, 2 different alleles. That's what heterozygosity is. And this is very, very important. Sometimes the allele can be dominant, and sometimes it can be recessive. In this example, I'm going to denote dominant as capital letter R, and then recessive as lowercase r. Okay? So in this case, if we have 2 dominant alleles R that produce the phenotype purple flowers, that means that only one dominant allele is required for this particular phenotype to occur. Okay, so there is a link between the dominant recessive characteristic of a particular allele and the phenotype. Okay, and so dominant means that you only need one allele, one dominant allele, for the trait to be expressed. You could have two dominant alleles, you're going to have the same trait. And then recessive, here we have a homozygous recessive, okay? And for the recessive trait to occur, you need to have the 2 copies of the allele, of the recessive allele. So if you have one copy of the recessive allele, you are not going to express the recessive trait. Okay? Any questions? Is it kind of clear? So here we have one dominant Y, which refers to yellow, capital yellow. Capital Y is yellow and lowercase y is green. So, what's the phenotype of uppercase Y, lowercase y? What's the phenotype? Done? Someone said green? I have it. How about lowercase yy? Well done. And capital Y? Fantastic. Now, there's a funny phenotype. Do you have detached earlobes or attached earlobes? Can you just check? Any attached earlobes? Mine is detached. Right, so detached earlobes are dominant, so it's a dominant phenotype, whereas attached earlobes are recessive, it's a recessive trait. Okay? So as you can see, this has clear, this just doesn't just apply to bees, it also applies to earlobes in humans. And like this, there are many other traits. Okay, so what's your ear phenotype? Can you raise your hand if it's attached? Wow, that's quite a lot. And then I guess the rest you are detached. Can you raise your hand if you are detach your lobe? Okay, cool. Yeah, so some of you are recessive, some of you are dominant. So for if you decide to have offspring, make sure that, depending on what type of offspring you want to have, you take into account your phenotype, just in case. Okay, so we use these terms, diploid and haploid. Okay, so two copies of the same chromosome, of all chromosomes, is called diploid, and we call them 2N. And a haploid cell, we call it N. Okay? So, in reality, so we humans, if we were to count every single chromosome we will have 46 chromosomes, right? But actually we have 23 pairs, right? And they're all in the nucleus of the cell. So meiosis. So this is just a reminder, because obviously all of our cells, all of the cells that we have in our body, they are diploid, right? So how do we stop from, if there wasn't the reduction through meiosis into a haploid gamete, then every generation we would be, times 2 the number of chromosomes. So this process basically explains how the reduction of chromosomal number into the gametes occur. And actually, it's an interesting process, because it's not just during meiosis, there's chromosomal shuffling. So it not only happens that the number of chromosomes is reduced to end up with haploid gametes, but also the chromosomes that you get the haplotype that you get in the gametes is actually being shuffled. So you're going to have in the gametes bits of chromosome that comes from the dad and bits of chromosome that comes from the mother of the person's gametes. Okay, so there's this process of generation of variation, which is very, very important, and it explains why, you know, we look like our parents but not exactly, and then sometimes there are you know, we're not an exact combination of your mother and your father. Okay, so that is explained by the process of meiosis. So so we have the fertilization, at which point basically you end up having a new cell, the zygote, where you have one chromosomal copy coming from the mother and the other one from the father. And then for every other replication that we have of the cell, we don't have meiosis anymore. Now we just have mitosis, where there is no reduction in the number of chromosomes nor any kind of shuffling between homologous copies of the same chromosome. Does that make sense? Any questions? Cool. So just to sort of insist that the only cells that are not diploid in our organisms are the sort of sexual gametes, where they have to be haploid, otherwise you will end up each generation duplicating the number of copies that you have for your chromosome. Okay, so also I must apologize that this it says there lecture 3, and then in the other slides it will say lecture 4, but this is kind of lecture 5, I understand. The reason is that I've just been last week in a trip looking into some communities in South America, try to understand their genomes, and I had to sort of change my class after I'm back to here. So, but it doesn't really matter. I think what really matters are these concepts that we're looking into. So we have two laws of inheritance: the law of independent assortment, and then the other law that we saw, the law of segregation. Okay, so the law of segregation means that the genes are going to be basically segregated, they're going to be separated, and independent assortment means that the alleles are going to be independently inherited. Back to the summary. We know what a gene is, which is a heritable factor that controls for a specific characteristic or function. And I also said that a gene is basically a segment of DNA that encodes for a protein. Although you can have genes that only encode for RNAs, but let's say just for now that. Then alleles are specific forms of a gene, forms of variants that differ by one or a few bases and occupy the same physical location on a chromosome. This is very important, okay? The alleles are always in the same place. Okay? So physically speaking, we know that for position 3,450,000, we have two alleles. We may have a T or an E, a T or an A, let's say. Then dominant alleles, they're expressed if they're present. So you only need one dominant allele for it to be expressed, and recessive alleles are only expressed when homozygous. that is when the two copies of the same chromosome have the same recessive allele. A genotype is the genetic makeup of the organism, and the phenotype is the appearance or traits that we are measuring and that are related to the expression of a particular gene. Any questions?
When we say homologous,
Homologous, yeah.
what do we mean?
What do we mean?
Yeah.
Yeah, we mean that we have the same allele. Well, we mean two things actually. Homologous in the sense of what a chromosome is. So you have the same chromosome. One is homologous to the other copy of chromosome 1 for the two copies that you have for each chromosome. And then you can have homologous. Well, actually, you have homozygous alleles. So I go back into what I said. So homologous only really applies to chromosomes. And when you are talking about alleles, then you are talking about homozygosity or heterozygosity. Thank you. Any other questions? Okay. So let's look at Mendel's laws of inheritance. Sorry, you mind if I just close that tiny gap there? We're just basically recapping from what we said earlier. So the interesting bit about P's is that they can be easily manipulated, as you can see here. So you take the estamens from a purple flower. The purple flower in this case is the dominant what? Dominant trait, dominant phenotype, right? Because dominant is applied to the phenotype. So purple is dominant, white is recessive. So if we cross purple with white, yeah, and then we get the first generation, they all going to be purple. Yeah? So then once we cross over between first generations, then the white recessive trait can come into view. So Gregor Mendel studied 7 traits. So seed shape, you have round, capital R, or wrinkled, lowercase r. Seed color, yellow, capital Y, or green, lowercase y. Flower color, purple. I mean, this here is sort of red, pink. So, but anyway, it's purple, which is the dominant, and white, which is recessive. Axial flowers or terminal flowers. Pod shape, inflated or pinched. Pod color, green or yellow, and then plant height, tall or short. I mean, again, I think just to be able to have this dichotomy between the different traits was something very, very lucky about or very, very useful about P's, because in reality, you know, you may have many different types of traits, like, you know, you can have like hair color. You may have like blonde or black or ginger hair. So it doesn't always follow this dichotomy, but for the purposes of understanding how Mendel's laws work, we're just going to focus on these sort of two traits, okay? But as always in biology, all the theory that we study has to be taken with a pinch of salt. But these patterns are very useful to understand for then applying it to genetics, medical genetics, for instance. So it was quite interesting that Mendel observed the same type of behavior for all 7 traits. So you have the first generation, which is the consequence of cross-fertilization. So you are crossing one trait with the other. And in terms of the type of the seed color, you always got yellow when you had the cross-fertilization between yellow and green. But then the second generation, which can be performed by self-fertilization, so you have the estamens and then you wrap them with the same plant, then you get the pattern of dominant recessive that we've studied, 75% yellow and 25% green. So Mendel proposed that inheritance of traits is determined by hereditable factors. So what are the hereditable factors in this case? How do we call hereditable factors nowadays? Nowadays, at that time the word gene hadn't been invented, but he called them hereditable factors. Okay? And he suggested that these factors come in alternative versions, which account for the observed variation in the traits. Okay? So we have a direct relationship between the chemistry of the nucleus and then of the DNA, and then what you observe at the level of the individual. And this is really something that we take for granted today, but the fact that we could assign traits to specific hereditable factors, I think that was an incredible discovery. Yes, so these factors are what we now call genes, and the variations what we call alleles. Variations at the level of the gene, okay, not at the level of the trait. Variations at the level of the trait is what we call the phenotypes. So for each trait, a diploid inherits 2 alleles of every gene, and 1 allele from the mother, 1 allele from the father. We know that already. So how are genes transmitted from parents to offspring? So we have these 2 laws, the law of segregation and the law of independent assortment. The law of segregation means that for 2 alleles in each gene, they separate when gametes are formed. Okay? And each gamete receives 1 allele for each trait. That's what segregation means. So the alleles are separated, 1 for each gamete. Okay? And the molecular process is through meiosis, where, as I said, we start with a diploid, then there is replication, meiosis 1 and meiosis 2. So in the end, you end up with gametes that are haploid. And interestingly, you're going to have chromosomes that are mostly coming from the mother and mostly coming from the father. But something which is not shown here is this process of shuffling that also occurs in meiosis, but which for simplicity we are not showing here. So in this case we have two copies of the same chromosome. One copy, this copy is actually, can you tell me, is it homozygous or heterozygous? We have two different alleles for the same position here, and that is heterozygous. Yeah? And that means that we're going to have gametes with different alleles, because the alleles are independently segregated. Okay? So the two alleles for each gene separate when gametes are formed, and each gamete receives one allele for each trait. So we have here a heterozygous P, and then the haploid gametes, they're going to have one allele which is recessive, one allele which is dominant, and one allele that is recessive. Let's look at Punnett square. Can I just say that it's likely, usually we get one of these in the exam, just in case. So maybe you might want to pay special attention. So what are these Punnett squares? So a Punnett square is basically where we just look at all of the possible type of offspring that you can have when you cross two sexually opposite organisms of the same species. And then it also tells us about the frequency of the different alleles that we're going to have and the different traits that we're going to have. So the Punnett square shows the probability of a genotype, and all allele combinations are equally likely. So let's then cross. We have a capital B lowercase b mother and a capital B lowercase b father. We see them there. Okay. So we just basically look at all the different combinations. So capital B, you get BB, lowercase capital B and lowercase b, you still get purple because it's dominant. Same here and same there. Now we have the two recessive lowercase b, and that's what makes the flower to be white. So if we have the genotype capital Y, then we have yellow. If we have lowercase y, we have green. So if we have capital Y gametes and we cross them with lowercase y gametes, they're all going to be, the first generation, they're all going to be homozygous, right? 100% yellow, because yellow is dominant and green is recessive. So, first generation. Now we're going to self-fertilize, okay? So all of them, so it's the same genotype. So we have capital Y and lowercase y for eggs, and capital Y and lowercase y for sperm. And this, we are talking about the second generation. So what are we going to have? 25% are going to be capital Y, 25% capital Y, lowercase y, 25% lowercase y, capital Y, and then 25% lowercase y. So again, the ratio 75% yellow, where only one copy of the allele is required for the yellow trait to be expressed, and then 25% green, meaning you need both alleles to be lowercase y to be expressed. Any questions? Is it getting a bit boring? Hopefully not too boring. Okay, so does the inheritance of one trait influence that of another trait? So in other words, does the seed shape affect seed color? Okay? So the second experiment is a dihybrid or two traits cross, where we basically follow the inheritance of two traits in the same cross. We call it the dihybrid cross. And the question is, are they inherited independently, yes or no? That's what he was trying to understand. So, and he was able to find that actually seed color is inherited independently of seed shape. And that's what we call the Law of Independent Assortment, where alleles of different genes segregate independently during gamete formation with the characteristics of 9:3:1 ratio. And that has been observed for all 7 traits that Mendel showed. So let's have a go at Mendel's second experiment. So here we have capital R, capital Y, and that produces rounded yellow, and then lowercase r, lowercase y produces wrinkled green. Okay, so let's see what happens. Now let's look at the proportions. So let's complete the Punnett square. So this is more like the type of thing that you would be required to perform on your own. So we have all of the different traits, all of the different alleles, and now we're going to see the proportions of the different combinations. So here in the top square we get rounded yellow. How about next one on the left? What do you think we will have here? Rounded yellow? Yes, very good. In the middle, what do you think we'd have? Rounded green, because basically what we have the capital R, and then lowercase y. Okay, so this is what it looks like. Brown is dominant, wrinkle is recessive, yellow is dominant, green is recessive. So we have 9 round yellow, 3 wrinkle yellow, 3 round green, and 1 wrinkle green. So Mendel found that P traits were either dominant or recessive. This is the law of dominance. And that traits did not become mixed. See, they actually remain distinct in later generations. So that was quite interesting. And that contradicted the scientific opinion at the time, where they thought that basically you get watered down. He didn't discover or even know about the existence of genes. And today we know that many inheritance factors are alleles. So these observations on inheritance are also called the law of dominance, which is the third law of Mendel. So we have the law of segregation, the law of independent assortment, and then the law of dominance. So these are the 3 laws for Mendel's. Please remember them, okay? Because this is very, very likely to be asked. So the law of segregation states that the 2 alleles for each gene separate when gametes are formed. Each gamete receives 1 allele for each trait. At fertilization, the zygote receives 1 allele for each gene from each parental gamete, restores the diploid state. And the law of independent assortment states that alleles of different genes segregate independently during gamete formation. Anyway, so there's a lot to process there, digest. Any questions? Please remember that this is kind of useful for your evaluation, should we say.
I have a question. So both this first and second experiments demonstrated the law of dominance. Is that true or false?
No, the law of dominance, I think probably mostly with the second. But I think the first experiment also, shows that you have a dominant and recessive. So the first one as well, I would say. Would you like to have a little break? Okay, cool. See you in 10 minutes then.